Hi, my name is Liz McDaniel, and this presentation is about what factors control our breathing, both when we are awake and when we are sleeping, namely the inspiratory and expiratory neurons within the medullary respiratory center in the brain, as well as the pneumotaxic and apneustic centers, which also help control respiration. This is for Human Physiology 2 with Dr. Waller at Grand Canyon University. So our breathing or respiratory control, as you know, is rhythmic. And while we have the ability to control our breathing, for example, when we speak or sing, we also must continue breathing while we're sleeping. And we also breathe even when we aren't thinking about breathing. So in some respects, the neural control of automatic breathing is similar to the control of cardiac rhythm because it happens even when we're not thinking. But in other respects, it's very different. This is because while it is automatic in many ways, we are able to easily override this autorhythmicity when we want to, such as when we're speaking, when we're singing, or when we're swimming underwater, but also when we need to, such as when we have to sneeze or cough. I want to add also that all three of these primary factors are influenced to varying degrees by levels of oxygen and carbon dioxide in our blood, so that homeostasis of these gases within our tissues can be maintained. I'm going to outline the primary centers of respiratory control, and then in the next few slides, I will discuss the specific mechanisms that control breathing in more detail. First, the primary respiratory control centers are seated within the brainstem, which is right here. If you look at the top here, you can see the pons. And within the pons lies what is called the pontine respiratory group. This group consists of the two areas called the pneumotaxic and apneustic centers. And below that, within the medulla, which is right here, are two more neural bundles called the ventral respiratory group or ventral respiratory center. Remember that ventral means front, and the dorsal respiratory group or center, dorsal meaning it's located behind or more towards the back. The dorsal and ventral respiratory groups are both connected to each other, and they also have neurons extending into the breathing muscles in the chest, which are the accessory respiratory muscles, the internal and external intercostal muscles, and of course our diaphragm. I'll have another diagram of those respiratory neurons later on. Also, just notice here at the top of the respiratory groups, just below the pons, is something called the pre-Botzinger complex. This little area plays a special role in respiration, so I'm going to talk about that as well. So I'm going to focus first on the medulla or the medullary respiratory centers. So when the dorsal respiratory groups, inspiratory or breathing in neurons, fire, they stimulate our external intercostal muscles and diaphragm, which causes inhalation. And then when they cease firing, these inspiratory or breathing in muscles relax, and this causes what's called passive expiration rather than a forced exhale to occur. Once these neurons fire again, expiration stops. So if you look right here, you can see that the dorsal respiratory center is connected to the ventral respiratory center. Now this ventral area remains inactive or dormant during passive quiet breathing. But when respiration demand is high, such as during exercise or other stressful activity, this area is stimulated by the dorsal respiratory group. The ventral respiratory group is summoned to increase depth and rate of breathing to accommodate for increased oxygen needs, as well as the release of carbon dioxide during expiration. Now above the medullary respiratory group in the pons lie the pneumotaxic and apneustic centers, which are collectively called the pontine respiratory group. We can think of these areas as the fine-tuning respiratory centers because they work to produce smooth inspirations and expirations. These centers provide a sort of checks and balances system so that our breathing remains calm and orderly. The pneumotaxic center directly influences the dorsal respiratory group in the medulla by sending signals to turn off, you can see that right there, inspiratory neurons in order to limit the length of time of an aspiration. And the apneustic center works in contrast or opposite the pneumotaxic center by preventing the switching off of inspiratory neurons and sort of boosting the drive to breathe in. And here in this image is a slightly more detailed pre-Bossinger complex. I'll try to circle that area for you. This complex is located in the upper head region of the ventral respiratory group, and it works in a similar way to the autorhythmic cells of the sinoatrial node of the heart. That is to say, neurons in this complex exhibit pacemaker-like activity with self-induced rhythmic action potentials, and this stimulation drives the respiratory neurons within the dorsal respiratory group. Now, if you're wondering how exactly the medullary and pontine respiratory areas know when to fire and how deeply to signal inhalation or expiration and for what duration, the answer is that there are many coordinating factors. First is the Herring-Brewer reflex, or Brouwer, I don't know how to say that. This is the reflex that occurs when our lungs sense being full to capacity, and it exists solely to prevent the lungs from being overinflated. It's brought about by special receptors that lie in the intercostal muscles called pulmonary stretch receptors. These receptors are activated when we inspire large volumes of air, generally greater than a liter, and they send signals back to the medullary and pontine centers to cease inhalation when our lungs start to feel too full. Also within the respiratory centers in the brainstem are the central chemoreceptors. These receptors surprisingly do not monitor the partial pressure of carbon dioxide, which is what I think most people would assume, in the blood. Instead, they monitor the carbon dioxide induced concentration or partial pressure of hydrogen ions. So when hydrogen ion concentration is too high, these central chemoreceptors activate the medullary respiratory centers to increase ventilation so that oxygen levels are improved and carbon dioxide is expired. And finally are the peripheral chemoreceptors. I have a picture right here for you. True to their name, these receptors do not lie in either the lungs or the respiratory centers in the brain, but they sit on the arteries extending from the heart. Namely the carotid bodies, which are here and here, which sit just at the base of the carotid arteries supplying the brain, and the aortic bodies, which reside on both sides of the aortic arch. Unlike central chemoreceptors, which are sensitive to hydrogen ion concentrations, these peripheral chemoreceptors instead monitor arterial oxygen concentrations. And in order for these receptors to signal increased respiration, arterial pressure of oxygen has to be reduced by more than 40% or below 60 millimeters of mercury. So due to this relative insensitivity, these chemoreceptors are generally thought of as a type of emergency response for when oxygen levels are dangerously low. And all of this information was brought to you by Human Physiology from Cells to Systems by Sherwood.